Next talk is from Charlotte Summers. Charlotte is a reader in intensive care medicine at the University of Cambridge and consultant on intensive care at Addenbrooke's. Charlotte has been member of the UK CTAP, which is a COVID Therapeutics Advisory Panel, and importantly for this particular talk is Chief Investigator for the HEAL platform, which is trying to identify therapies for long COVID. So Charlotte, thank you.
Brilliant. So thank you very much for asking me to come and talk at this really fabulous symposium today. I'm Charlotte Summers from the University of Cambridge, and the bit that I'm going to talk to you about is finding therapies for post-acute COVID and the HEAL COVID platform trial. My conflicts of interest, just so that we're clear at the beginning, are that work in my research group is funded by a variety of sources, including GlaxoSmithKline and AstraZeneca, but none of that funding relates to COVID research. It actually relates to my long-term research interest in acute lung injury. And I've had consultancy fees relating to COVID from the people that you see here. But my most important conflict here is, as Mania said, I am the Chief Investigator of HEAL COVID, which is jointly funded by the NIHR and the Cambridge Biomedical Research Centre. So if 20 people get SARS-CoV infection, actually only a small number of them will get severe infection. And by severe infection, I mean infection that requires them to be hospitalized. The majority of them will be asymptomatic or have illness, which is something that means that could be managed at home in the community. Of the people that get admitted to hospital, only 1 out of those 20 will develop an organ failure that needs intensive care. And the COVID illness itself, as I'm sure you've discussed, but I think it's helpful to recapitulate at this point, has several phases. The kind of pre-acute phase, where you're predominantly asymptomatic and where there's a clear antiviral responses predominate. The acute phase, which is a combination of both antiviral and host immune response, and also a post-acute phase, which is probably most likely to do with the host immune response. Now, there's a lot of been known about host immune responses and the long-term consequences of respiratory infections for many, many years. In fact, probably for a century now. We've known with community-acquired pneumonia, people used to go and live by the seaside for up to 6 months after their initial illness, because that's how long the symptoms took to resolve. But when it comes to COVID, there's been some more detailed and granular data. So what are the potential outcomes? Well, when you have an acute COVID infection, you can recover, you can have persistent illness, and you may not survive. But actually, it's more complicated than that. There's these distinct phases that I talked about, where there's the pre-acute asymptomatic phase, which is unusual actually for a lot of acute infectious illnesses. Your acute phase can happen in the community, in hospital, or in fact in an intensive care unit. And then everybody has a variety of outcomes they can have in the post-acute phase. So whether you're in intensive care or the community, actually you can still have persistent symptoms, or you may resolve. Same for hospitalized patients. And when it comes to the critically ill population, I know as a respiratory trained intensive care consultant that actually for many years now, for the best part of a decade, we've recognised that our ICU patients can develop persistent critical illness. And in fact, actually having long-term clinics to follow up these patients has been a requirement for intensive care units in the UK for nearly 10 years. So what data is there? We were concerned to see data emerging from the United States, from the VA network and the Blue Cross network in the second half of last year, suggesting that there was a significant problem in the proportion of patients being re-hospitalised and dying in the 3 to 4 months after they left hospital. And then these data that came out initially as a preprint, but more latterly in the BMJ from researchers from the Office for National Statistics and others who are on this call, looked at 47,500 just over patients who were hospitalised and survived discharge from the wave 1 in spring 2020. Of those, 29.4% of them were readmitted to hospital in a median of 140 days. 12.3 of them were dying during that 140-day median time window after discharge from hospital, and 58.3% of them were surviving, not readmitted to hospital over that same time point. This was really striking. We were saying that a third nearly of patients who survived hospital discharge with COVID were getting readmitted in the 3 to 4 months after their hospital discharge, and a disproportionate number of those patients were dying. This was based on English patients, so we had a look when we were thinking that perhaps we ought to be doing something about this at what was happening in Scotland. And in Scotland, in wave 1, actually 5,392 patients were admitted to hospital, of whom 4,167 survived to discharge, and 12.9% of the people who survived to discharge died in the first 6 months after hospital discharge. And 58.3% of them were still alive at 6 months post-discharge. So those proportions of people who were dying post-hospital very much seems to align with both the data from England from wave 1, but also with that reported from our American colleagues. So what were the patients who were surviving, what was happening to them? What did they look like? Well, here FOSP was very helpful. And they looked at 1,000 patients just over who survived 5 months post-hospital discharge on average. And at that point, 29% of the patients who came to clinic at 5 months felt that they had fully recovered. 20% of them felt that they'd got a new disability. And 19% of them had by that point a health-related change in occupation. And the kind of post-acute COVID syndromes that people were describing in those first few weeks and months after hospital, and this is described also in patients who had never been hospitalised, but the focus of HEAL COVID is the hospitalised population. People very much complain of fatigue, joint pain, fever, respiratory symptoms, including predominantly shortness of breath, cardiovascular complications, neurocognitive complications such as brain fog, neuritis, loss of taste and smell, gastrointestinal symptoms, skin rashes and other dermatological manifestations, and urogynecological symptoms. So there's almost every system of the body is being impacted by having had an acute illness, including COVID. The ONS data was also striking for the fact that actually they looked at the number of people who were having new diagnoses after discharge to hospital. 29.6% of patients in that group were reporting a new respiratory diagnosis had been made after they left hospital. 5% nearly had a new cardiac diagnosis, and that was a major cardiac event, so the MACE classification. And nearly 5% had a new diagnosis of diabetes. At this point, we were very, very clearly of the view that it wasn't ethical to continue to sit and watch and study the natural history of this when there were probably interventions that we could deploy into clinical trials very much in the way that recovery had done for the hospitalised patients, because there were things that we could plausibly say may well alter the clinical outcomes. And so this led to us setting up, helping alleviate the longer term consequences of COVID-19, HEAL COVID, which is an urgent public health classified national platform trial, which, as I said, was funded by the NIHR earlier this year. So the objectives of HEAL COVID were to determine whether interventions in the post-acute phase of COVID-19 in hospitalised patients can improve the longer-term mortality and morbidity outcomes. The secondary objectives are that we want to see whether we can determine and evaluate treatment-specific and, most importantly, patient-reported outcomes and their response to interventions, and also assess the cost-effectiveness of the treatments that we're studying. So what are the population we're looking at? They're adults 18 years or over. They're hospitalised patients that are approaching the end of their index admission with COVID-19. So they're people who have been admitted for a primary SARS-CoV-2 infection-related disease at the point that they've come through the worst of their hospital stay and are getting ready to leave hospital. and they have to have SARS-CoV-2 associated disease as the reason for that hospital admission. So an overview or schema of the trial as it's currently set up: patients are recruited in the 5 days there or thereabouts prior to going home, and they're randomised to usual care, to apixaban 2.5 milligrams twice daily for 2 weeks, or atorvastatin 40 milligrams once a day for 12 months, and then a third active arm that is currently in setup. It's an adaptive clinical trial, as many of the platforms you'll have heard about in COVID are. And so we had planned interim analyses at particular event rates. And the primary endpoint is hospital-free survival at 12 months, with a range of secondary endpoints that I'll talk a little bit more about in a minute. So how did we get the interventions that we're testing? Well, thankfully, there, as many have mentioned, is a thing called UK COVID Therapeutic Advisory Panel, who are a group of experts that oversee a series of specific domain sub-panels, of which Manir is one, that review a huge variety of medications to determine in what setting and what types of therapies may impact the clinical outcomes from COVID. Very early on, it was suggested by NICE and others that there was a real question around what to do in terms of post-hospital anticoagulation in patients with COVID, because there were reports of an increased rate of venous thromboembolic events in the early weeks after discharge. And so apixaban, the oral anticoagulant, was felt to be important to address that. And 2 weeks was the therapy duration that CTAP suggested, because actually that's the point at which the risk from bleeding is probably thought to exceed the risk from thromboembolic events after discharge from hospital. Atorvastatin, which many of you will know for its lipid lowering actions, actually is being used because, based on work from the University of Birmingham and from Belfast, from people like Liz Sappi and Danny Macaulay over many years, both in community acquired pneumonia and acute respiratory distress syndrome, it's been known that statins have vascular inflammatory modulating effects, and thought that, as well as their cardiovascular secondary modification, those may well be helpful in the setting of COVID. So atorvastatin 40 milligrams once a day for a year. So what are the outcomes we're looking at? The primary outcome is hospital-free survival at 12 months. We had a lot of discussion with regulators and others about the endpoint. Interestingly, the peer review bodies and initially the regulators felt very strongly that all-cause mortality was the appropriate endpoint. But actually, as part of Heal COVID, we've had extensive public and patient involvement and engagement, and the patients argued very strongly that actually, for them, having been hospitalised once with COVID, recurrent hospitalisation and the duration of time they spent in hospital over those 12 months were as important to them in terms of impact on their quality of life as death. And so they advocated strongly that we had hospital-free survival rather than just mortality. The secondary endpoints are all-cause mortality, hospital readmission after discharge, suspected serious adverse reactions, and patient-reported outcome measures, as well as incremental cost effectiveness based on QALYs. So what are the patient-reported outcomes? We're looking at measures of fatigue, the modified MRC dyspnea scale, as modified by Alison Tong and colleagues in their COVID core outcome set, the COVID core outcome measure for recovery, EQ5D for quality of life, some other neurocognitive assessments, as well as assessments of the tolerability of the interventions and specific symptoms. The key thing was that we were trying to strike the balance between what is reasonable to ask of patients recurrently over time who have just been discharged from hospital and who are potentially suffering with things like brain fog. And again, here the PPIE was so helpful in shaping the trial, because they were really clear with us that actually anything more than about 10 minutes worth of questionnaires and data collecting in one go was intolerable. And what they didn't want to be doing was being required to spend large amounts of time repeatedly undergoing tests and answering questionnaires, because they felt it was quite burdensome. So we tested out extensively our panel of patient-reported outcomes and the means in which we collect them, that I'll talk a bit more about in a minute, in patients both who were more late in their long COVID, but also in patients who were actually in the post-acute phase. So we had tremendous support from the patient communities. We are at the point now where we were funded on the 8th of March. Our regulatory approvals were all issued by the 8th of April. Our first site opened to recruitment on the 7th of May. Our first patient was randomised on the 19th of May, and recruitment continues. We are open at 90 sites across all 4 devolved nations in the UK. The red ones are ones that are currently doing their training, and the green ones that are ones that have already randomised. And as of this morning, and it goes up all the time, we'd randomised 411 patients and have 20 sites and set up, although it's not 89 anymore, it's 91, because 2 more opened over lunchtime. Delivering a trial such as this at scale in faster time points than we would normally requires a huge team effort. And I tried to show this by listing just some of the very many people that have contributed to Heal COVID. So the trial management group, the Liverpool Clinical Trial Centre are the people who operationally run the trial. We've got a Cambridge-based central clinical trial team. So all of our patient-reported outcomes, we offer patients the opportunity to tell us about how they're getting on and answer those questions via a digital platform from Apparito called Atom 5. But we know for a variety of reasons, including digital poverty, that not everybody has the ability to do that. And so the other option is that we can phone you and do those questionnaires on the schedules required via the telephone. We've also worked quite hard to ensure that the entire trial is delivered end to end in at least 7 different languages. So whether you do the app, the telephone, all of the consent processes, all of our patient-facing documents are available in multiple different languages to try and reduce the barriers to participation. In reducing some of those barriers, we've had huge support from the NIHR CRN, and Priest is our local lead, but also centrally from Izzy Baker and Nick Lemoyne. The Department of Health have been tremendous, as have NIHR, in making sure that any obstacles that can be removed to taking part in this trial are, including things like how do we stop there having to be prescription charges for patients taking part in research who are out in the community. All sorts of small obstacles to doing big scale research in a pandemic, DHSC have worked very hard to help us overcome. If you want to find out more about Heal COVID, you can look at our website or our Twitter feed. And actually, we've got a growing YouTube channel where we've put some of our educational content and talks relating to the trial from some of our trial management group. And if you want to contact me, because you've got some follow-up questions, you can get hold of me via either of these 2 routes too.
Thank you very much, Charlotte. That's great. Okay, open for any questions to Charlotte. Charlotte, maybe I can start off where people think of questions. How many arms do you think you might be able to open as part of this? You know, is an adaptive trial, as you said?
So the trial is designed that we can have 3 active interventions plus usual care open at any one time. And we've got stopping rules for futility and benefit for when we do the interventional arms. So if when we get to a review point, we find that, I don't know, apixaban really looks like it's doing absolutely nothing and that further recruitment is not going to be helpful, we can stop that and replace it with another intervention.
Okay, thank you. Mark Lazarus.
Hello, it's me again. Thanks, Charlotte. This looks like a really great trial. But I'm just noticing, all the trial, the main people in the trial committees, and didn't notice a rheumatologist. And the only reason I just sort of mentioned that is that I've just left the NHS and was doing a kind of a general rheumatology clinic, and was seeing a small number, you know, in my clinic just trickling through of unusual non-respiratory post-COVID type syndromes, you know, people with painful fingers, people with painful toes, brain fog, all sorts of and one very unusual case of a sort of a kind of a pancreatitis, actually, a sort of unusual pancreatitis and GI symptoms, but they didn't start with respiratory symptoms. And so I just wonder whether you've got a particular arm that maybe or sub-analysis that's going to look at patients that don't present with respiratory illness.
So remember, we're recruiting hospitalised patients regardless of what they present, and so they're being recruited at the point of their hospital discharge. We're not recruiting patients from clinic.
Okay.
Other studies that are set up very differently, and I think Amajit is here, he can potentially talk to you about Stimulate, that are based for community COVID. We're very much about the patients who were hospitalised for their primary COVID, trying to decrease the number of people who die and get readmitted over the first 12 months. But absolutely, there's a plethora of people who are presenting to different clinics. Some of our ICU patients are presenting to the standard ICU follow-up clinics that we've always had, with all sorts of manifestations.
Okay, yeah.
Thank you. Julie.
Great, thank you. Thank you for that, Charlotte. That was great. Really interesting to hear about patient and public involvement and how that really shaped the protocol. Great example. I was wondering what your plans were for sort of ongoing engagement now you're up and running, and how that's working.
So we've got an actual PPI team embedded. So Paul Wicks leads on PPI, along with Amanda Schranks, who's the NIHR Cambridge Biomedical Research Campus lead for PPI. And we've got a series of people who have helped us from different demographics from across the country, because obviously having inclusive PPIE rather than your kind of go-to group that you might find easiest to engage with are really important. So we've got people that we've engaged with so far from Cambridge, from Oxford, from Liverpool and from Birmingham. But in addition to that, we're putting together a formalised patient panel as part of the study.
Thank you.
The other thing to say, I guess, is dissemination plans. We've worked quite hard, not just on what the trial looks like, but also in how do we communicate what we find out about in our trial, as well as how the trial's going with various different audiences, because what might communicate well to a bunch of academic researchers may not be what the patients or even other care providers want to know about. So we've tried to think quite a lot about our comms, hence we've been trying to do things like YouTube channels, Twitter feeds, talking to public groups about the kind of work we do in various different ways. So having advice on good ways to do that that are meaningful to the communities we're trying to impact has been really important to us as well.
Thank you. We've got about 5 minutes before lunch break, Charlotte, so maybe I can start off with a more kind of forward-looking question, if you like. And what I'll do is, after that question, is bring in Amitava Banerjee in as well, and he can tell people about the trial that he's undertaking. So Charlotte, first of all, we've had a discussion about this briefly and so on, about your thought. You heard the complexity of all the manifestations which are occurring, multiple systems being affected and so on, and how you feel might be best way to be able to stratify patients for developing trials for the future, so that we can actually, because if you put every patient into the same arm, you're going to need a very large trial with because the effect size is going to be quite small, but stratifying may actually allow you to be able to do smaller trials with better effect sizes.
This is an issue we have struggled with in intensive care for a long time. And the reason that we struggled with it in intensive care is we're essentially medicine for syndromes in the ICU. We have sepsis, we have acute respiratory distress syndrome, we have post-critical illness. We're not really a specialty that deals with diseases so much. And so finding ways of effectively pre-stratifying your patients at the point of care in an acute illness is actually quite challenging, which is why very few people have managed to do it. Even in cancer, actually, the number of trials that have managed prospective stratification are more limited than people might imagine. I think it's important to make sure that there's alignment between the endpoint that you're measuring in your trial, the intervention you're testing, and the way in which you stratify. So there are ways, and the setting in which you're stratifying is also important. For me in ICU, it may be fine to say I'm going to stratify on, I don't know, PaO2, FiO2 ratio because I always have a blood gas. However, that doesn't work in a community setting because they don't have blood gases. And so actually validating the things you stratify on are quite important. We looked in the example of hypoxia, whether we could stratify for some interventions within HEAL COVID by looking at oxygen saturation versus the amount of oxygen you were receiving at the time, and tried to validate that using the ISARIC data set as to whether, if we stratified you by your SF ratio, did you have a different outcome even at 3 months? And actually we couldn't validate that stratification, and so we were unable to take that forwards. So I think the short answer is that actually it's more challenging than people think. Biomarkers historically have not stratified well for intervention. So measuring things like IL-6, as you will know, and more recently actually for anticoagulation, measuring D-dimers, which we were all absolutely convinced would tell you how much anticoagulation to give to your patient with COVID, haven't worked at all. So we know less than we think, but validation I think is really important.
Okay, thank you. Ami, I don't know whether you want to come in and just to let people know about your big study that you're undertaking, including the drug platform and so on.
Thanks very much, Munir. And I couldn't agree with Charlotte's last comments more, actually. I think that they're particularly relevant in long COVID, that we would like to move towards stratifying, but we're early on the journey, and even compared with D-dimer and other markers that we've given a go in clinical medicine. So I'm based at UCL, and I am leading a consortium with colleagues from over 20 different institutions called Stimulate ICP, funded in the recent call by NIHR for long COVID interventional studies. And so we have 3 things that we're doing really. But firstly, we're looking to evaluate current care, and as Kieran Collison showed in her talk, that's a moving feast at speed across lots of different sites. So we're trying to do that, and we're particularly focusing on 6 centres to start with. That's Liverpool, Hull, Derby, Leicester, Exeter and London. And the second thing we're doing is a drug trial embedded within a cluster randomized trial. And so if I take the cluster randomized trial first, that is looking at both a specialized investigation called CoverScan, which is a multi-organ MRI scan, versus usual investigations in the non-hospitalized long COVID clinic. And at the rehabilitation stage, we have a digital enhanced rehab platform called Living with COVID Recovery, which we're comparing with the NHS recommended pathway, which is Your COVID Recovery. So both app-based rehab. And then within, and that's randomized at the primary care network level. Embedded within that is a drug platform, and the exact drugs in that trial are being decided at the moment. And this would be a project over 2 years with 3-month outcomes. The primary outcome is actually fatigue, and I can speak to how we've arrived at that as the primary outcome. And the third piece of work we're doing, which speaks also to Kerry Collison's last part of the talk, is about inequalities and looking at how we can address gaps and inequalities in referral to long COVID clinics and learn from that for other long-term conditions.
Brilliant. Thank you very much. That's a great summary of your trial. So if there's one more question, so surely last question before we break for lunch.
Yeah, thanks. I'm not sure if it's a question or more of a reflection that we could maybe take into lunch and some of the discussions this afternoon, but what struck me from this morning is the complexity in a very difficult, extraordinary environment. So we've got, you know, the disease itself and trying to understand that, any effects of treatment, but then also what are the effects of lockdown, for example, sedentary lifestyles, mental health effects. So how do you tease that out? There's something about the longitudinal perspective here, and I'm thinking of, that cohort of women in their 40s and 50s who'll be hitting perimenopause, and how do you start to tease that sort of thing out? And that's just one example. And then the other bit is any impacts of delays of treatments or elective waiting lists and things like that, and how you tease that out. And I mean, it's been brilliant to hear how people are starting to tackle the question, but it's definitely, the more you think about it, the more complex it seems to get.
Thank you, Shirley. That, yes, you're highlighting the complexity and additional aspects that we need to think about. So really important. So thanks very much for that. So definitely need to take that back into the breakout groups later on. So we are going to break for lunch, but please do come back, listen to David Jeffries, and then do help us with the breakout groups where you can have a discussion and really input into what your thoughts are in this area, and so that we can actually learn much more from each other about what is going on, where we need to move forward. So thank you very much for your talk, Charlotte, and for the talks this morning. Really great talks all this morning, and so on. So look forward to the next talk and then the discussions in the breakout groups.